Hello students, welcome back to my biology online classes. Today I am going to discuss with you about signaling receptors. In my previous classes you have already known about this conception of receptors in unit 7 topic cell signaling. So now let's discuss about signaling receptors. Signal are given by that cells which receive signal from its surrounding, and they can secrete ligands. They secrete ligand molecules. Ligand molecules will go to target cell, and target cell, all of you know, there are two types of receptor. In target cell there are two types of receptor. In my previous classes I have already discussed. Signaling receptors are of two types: intracellular receptor and cell surface receptor. Intracellular receptors are in a cell, either nucleus or cytosol. So, intracellular receptors are present either in nucleus or in cytosol. Got it? Normally receptors are chemically protein or glycoprotein in nature. It binds to signal molecules. Ligand bind to receptor. What happens? A conformational change will occur. Conformational change, a structural change will occur in which? In receptor. Okay. Conformational change will occur in receptor after binding ligand, with ligand. The structural change of receptors are observed, and this conformational change of receptor that initiates a sequence of reaction, and this leading to a specific cellular response. So when ligand bind with receptor, a conformational change is observed in receptor, and this conformational change in receptor, it initiates a sequence of reactions inside the cell, for which the cell have shown a specific cellular response. Now come to the intracellular receptor. Intracellular receptors are also of two types: type 1 receptor and type 2 receptor. Type 1 receptors are steroid hormone receptor, and type 2 receptors are present in nucleus. And example, vitamin D3 and thyroxine hormone receptor. So normally intracellular receptors are located inside the cytosol or nucleus, and extracellular lipophilic signal molecules can easily pass the PLP layer of plasma membrane, and they can bind with intracellular receptor either present in nucleus or present in cytosol. Example, steroid, retinoid, they are spontaneously diffused through plasma membrane and bind to intracellular receptors present in the cytoplasm or nucleus. Within the cell, what happens? The intracellular ligand receptor complex controls the activity of responsive genes. Ligand receptor complex controls the activity of specific genes. For this, transcription initiates. So take an example. Steroid hormone, the most common receptor. The cytosolic steroid hormone binds with cytosolic receptor proteins. Then conformational change occurs, and they bind to DNA sequences, which are known as HRE or hormone response elements. And the next step, they can activate transcription of responsive genes. In many cases, this response, it may take two steps: primary response and secondary. Primary response and secondary response. What occurs in primary response is direct induction of transcription of a small number of specific genes within about 30 minutes, very fast, okay. And the secondary response product of these genes means primary response gene products, they can now activate other genes, got it, and produce a delayed secondary response. So these are all about intracellular receptor. Now let's concentrate about some extra information about type 1 receptors. Type 1 receptors are all steroid hormone receptors. Steroid hormone receptors. Example, glucocorticoid receptor. This glucocorticoid receptor is present in cytoplasm, complexed with a heat shock protein 90, HSP 90. Now this glucocorticoid receptor plus HSP 90 heat shock protein, okay. Normally glucocorticoid receptor is remain bind with HSP 90. Normal, it is in their normal condition. Now glucocorticoid ligand is coming, conformational change occurs, it dissociates from HSP 90, then move into nucleus and activate genes. Gene activation occurs. Glucocorticoid response element. So this is all about type 1 receptor with one example. Now let's concentrate with type 2 receptor. Type 2 receptor present in nucleus. Type 1 receptor is present in cytosol and type 2 receptor is present in nucleus. One is receptor for vitamin D3 and thyroxine. Now type 2 receptor plus response element. RE means response. Normally this one is normal condition. Prior to ligand binding, type 2 receptor remain bound with RE, okay. In the absence of ligand, normally in the absence of ligand what happens? Receptor can repress transcription, normally. But in the presence of ligand, receptor can stimulate transcription. In the presence of ligand, transcription initiation occurs. Thus, the same protein acts as activator or repressor. When binds with ligand, they can initiate, activator act as activator, and in normal condition when binds with RE, in absence of ligand, prior to bind with ligand, they can act as repressor, and they belong to nuclear receptor family. So it's all about intracellular receptor. Now come to the next point, cell surface receptor.
Now let's discuss about another type of receptor, cell surface receptor. Means this receptor is present on the outer side of the cell on plasma membrane. There are mainly three types of cell surface receptor. First one is G protein coupled receptor. Second one is ion channel receptor, and the third one is enzyme linked receptor. So there are three types of cell surface receptors: G protein coupled receptor, ion channel receptor, and enzyme linked receptor. Among these three types of cell surface receptor in your syllabus, there are G protein coupled receptor and enzyme linked receptor. Not ion channel receptor is included in your syllabus. Enzyme linked receptors are of two types. First one is RTK, and RTK means receptor tyrosine kinase. Second one is tyrosine kinase activated receptor. Example of tyrosine kinase activated receptor is JAK-STAT, which is in your syllabus and very much important. In case of RTK, tyrosine kinase self-act as enzyme-linked receptor, and in case of tyrosine kinase activated receptor, Here, tyrosine kinase is not receptor itself, but it associated with some another receptor. Tyrosine kinase associated, attached receptor, and the example is JAK-STAT. Which one is in your syllabus? There are so many types of RTK or receptor tyrosine kinase. Okay. First one is RAS-MAP kinase pathway, which one is very much important and also in your syllabus. MAP kinase pathway and IP3DAG pathway, PI3 kinase pathway and insulin signaling pathway. Let's sum up cell surface receptor. Cell surface receptor is of three types: G protein coupled receptor, ion channel receptor and enzyme linked receptor. And enzyme linked receptor are of two types: RTK, receptor tyrosine kinase, and tyrosine kinase activated associated receptor, which example is JAK-STAT, and it's very much important. Receptor tyrosine kinase again divided into RAS-MAP kinase pathway, MAP kinase pathway, IP3DAG pathway, PI3 kinase pathway and insulin signaling pathway. Out of these 5 types of RTK, RAS-MAP kinase pathway, MAP kinase pathway is in your syllabus and very much important, and resting 3 IP3DAG pathway, PI3 kinase pathway and insulin signaling pathway, they are also major types but not included in your syllabus. Now let's concentrate about the different types of GPCR, G protein coupled receptor. First of all, GPCR and G protein. Second one is CAMP and PKA, protein kinase A. Third one is protein kinase and phosphatase. Fourth type is GQ and phospholipase C beta. Fifth type of GPCR is calcium calmodulin complex, which one is also very much important. Next one is rhodopsin and GT. And the last one is about GPCR adaptation. So the GPCR is about total 7 types. So these are all about in detail cell surface receptor, all about intracellular receptor and their types, and also types and examples of signaling receptors according to your syllabus.